Hello, good morning students. My name is Avantika Pandey and I am going to teach you a topic from biotechnology, that is from 11th and 12th standard, and the topic is transgenic plants. Okay, let us first know what are the transgenic plants. Transgenic plants are the ones whose DNA is modified using the genetic engineering techniques. Transgenic plants are the plants which are being in which the new DNA is being introduced into the plant, and they are genetically engineered to produce a better plant by which we can improve its nutritional factors, its resistant factors, etc. So these are called as a transgenic plants. Now the whole thing which we are going to know about the transgenic plants are here given in this PowerPoint presentation. A new trait to the plant which does not occur naturally in the species. In these transgenic plants, a new DNA is being introduced into the plant and it is called as a new trait, and they are being they are introduced into the plant but they do not occur naturally in the plants. So we have to introduce these uh, this particular DNA into the plant, and this inserted gene sequence is known as a transgene. This particular DNA which we are going to introduce into the plant with the help of the genetic engineering is known as the transgene. Okay, now what are the advantages that we get from these in the genetically engineered plants, that is transgenic plants? The first advantages are these improve the shelf life, higher yield, improve quality, pest resistant, tolerant to heat, cold and drought resistance, and help in the against a variety of biotic and abiotic stresses. Abiotic and abiotic stresses are the weather conditions, and the abiotic stresses are the for example pest, virus control, etc. Okay, now moving on to the next slide. Now we are going to learn how are we going to develop these transgenic plants. First of all, we should know the method by how we can develop these transgenic plants. These genetically engineered plants are generated in the laboratory. First, they are generated in the laboratory by the help of the recombinant DNA technology, which is also known as the particle gun method. So both this method are the same method. This is known as a recombinant DNA technology method, or also the particle gun method. This is done by the help of a specific bacteria which is known as Agrobacterium tumefaciens. These are the soil dwelling bacteria and they are mostly found in the soil. They help in the nitrogen fixation also. You must have heard about the nitrogen fixation. So these are the bacteria which help in the nitrogen fixation, and here also they are very helpful in the development of the transgenic plants. Okay, now we are moving, first we are going to know what is this gene gun method and what is this recombinant DNA technology method. In this, the DNA is bound into the tiny particles of gold or tungsten. The particular DNA is introduced into the gold or tungsten particle, and then it is bombarded into the particular bacterium plasmid, through which the DNA is being introduced into the bacterium DNA. So let us know what is actually this whole machine, this whole mechanism is all about. We can learn it with the help of this diagram. The diagram makes it very easy. What is the recombinant DNA technology method and how it can be and how it can help to generate the transgenic plants. Firstly, this is the agrobacterium. We can see that this is the Agrobacterium tumefaciens cell, and this is the particular plasmid. In this plasmid, the bacteria has its own particular plasmid. So in this plasmid, we are going to introduce the particular DNA, the new trait of DNA, which is not naturally present in the bacteria. We are going to introduce this bacterial DNA into the plant DNA. Okay, now this is known as a TI plasmid. This is the restriction site. Here we can see that this is the restriction site. Firstly, we will take the desired DNA containing gene for desired trait. This is the DNA which we are going to introduce into the TI plasmid. So this particular DNA gene, insertion of gene into the plasmid using restriction enzyme and DNA ligase. So this particular gene is going to be introduced in this TI plasmid, and they are being this is done with the help of the two enzymes, that is restriction enzyme and DNA ligase. Restriction enzyme are the enzymes which cuts the plasmid. It cuts the plasmid at particular ends, at specific ends, and DNA ligase helps to bind that particular DNA into the TI plasmid. Now we can see in this diagram that the recombinant TI plasmid has been created, and with the help of the DNA ligase, this particular new segment of DNA is being introduced into the new plasmid. Now this is the recombinant DNA plasmid. This is called as the recombinant DNA plasmid. Now this plasmid will be introduced into the plant cells with the help of the tissue culture technique. This is the plant cell, this is the chromosome, this is the tDNA carrying the new gene within the plasmid chromosome. This is the new gene which the plasmid is carrying. This particular plasmid, this particular DNA will be introduced into the plant cell. Now this plant cell, after the tissue culture development, after the tissue culture, firstly we will keep it in the lab in the incubator at optimum temperature and humidity, and after that this plant will be regenerated. This whole plant will be regenerated, and this plant will be called as a plant with a new trait, that is the transgenic plants. Okay, now how to make a transgenic plant? As we have already explained in the diagram previously, that the new DNA is being introduced into the cell of the plant. So firstly what we have to do is isolate the DNA that codes for the protein you want to express. Firstly we have to take the DNA which we want to, whatever protein we want to express in the plant, we want to take that part of the DNA. So this segment of DNA, that is ATGGCA, here as we know, as we have already studied in class 11 and 12, that A codes for T, T codes for A, G for C, G for C, C for G, and A for T. So this is the particular plasmid. Insert the DNA into a plasmid. This particular plasmid will be introduced into the plasmid of the bacteria. This is the broth of the bacteria, we can see over here. Now insert the plasmid into the bacteria. Grow a large amount of bacteria containing this plasmid. Once the plasmid is being introduced into the bacteria in this broth culture, now when the cells will divide, the plasmid will automatically divide, and this will contain a large amount of plasmid carrying the particular nutrient. Okay, now dip the flowering plant into a large amount of bacteria. So whatever the plant we want, that particular flowering plant will be dipped into this amount of bacteria, and this gives the bacteria the opportunity to insert the DNA into the plant cell. So the bacteria will automatically insert the plant cells into itself, insert the DNA into the plant cells. So the plant cells will automatically take these bacteria, generate into a large number of plasmids, and then with the help of this we can develop a new transgenic plant. Now Agrobacterium tumefaciens has a natural ability to insert new DNA into a plant genome. Why this particular bacteria is used? This particular bacteria is used because it has a natural ability to insert the new DNA into the plant genome. It eases the process of transferring the new DNA into the plant's genome. The DNA that agrobacteria naturally insert causes changes in a plant that result in the formation of tumor at the infection site. Now the DNA that agrobacteria naturally insert causes changes when this agrobacterium bacteria, it introduces the particular DNA into the plant cell. At that point of transfer, that point is called as the infection site. At that infection site, a tumor is formed at the infection site. The tumor produces nutrients that the bacteria need. So at that particular site, the transfer occurs. That is, the bacteria transfer its DNA into the plant cell, and that at that particular position, a tumor is formed. With the help of tumor, the bacteria gets the nutrients. It provides the bacteria have special proteins that allow them to evade plant defense systems and insert DNA into the plant genome. So because of that tumor, the tumor creates special proteins that allow them to invade the plant defense system, to evade the plant defense system and insert the DNA into the plant genome. By the help of this, because of this, agrobacterium tumefaciens is particularly used in the recombinant DNA technology. The agrobacteria insert this new DNA into a huge number of cells. Some of these cells are cells that form seeds which will grow into the new plants. Now when this bacteria inserts its new DNA into the large number of plant cells, some of these cells are grown and they form seeds which will grow into the new plants. These particular cells, when they will be sown in the soil, they will develop into a new transgenic plants with a new trait or new character. Okay, now what are the types in between this recombinant DNA technology can occur? So there are different types of kingdoms and species in which this particular recombinant DNA technology can occur very easily. So first is the species within the same kingdom, that is plant to plant. Like plant belongs to the same kingdom. So if the DNA is being transferred from plant to plant, it will be great to create a new genetic plant. So it can occur between plant to plant. Then it can occur between kingdoms, that is bacteria to plant. Okay, that is another category of cisgenic plants. Cisgenic plants are made up of using genes found within the same species or a closely related one where conventional plant breeding can occur. So cisgenic plants are the plants that are made up of using genes found within the same species. These are the plants which have the genes within the same species or closely related ones. So in this also the recombinant DNA technology can occur easily. Okay, some breeders and scientists argue that cisgenic modification is useful for plants that are difficult to crossbreed by conventional methods. So in this process, if cisgenic plants are available, so recombinant DNA technology can occur very easily as compared to the conventional methods like crossbreeding, etc. Okay, now what are the advantages of these transgenic crops or plants? Firstly, herbicide resistance. These plants can be herbicide resistant. As we know that herbicides have a lot of side effects on plants that we eat, we consume. So the advantages of these transgenic plants are they can make the plant herbicide resistant. They can tolerate the herbicides. So here are some of the examples through which the plant can resist the particular herbicide with the help of a particular gene. So one example is glyphosate resistant transgenic tomato, potato, tobacco, cotton, etc. are developed by transferring ARO gene into a glyphosate synthetase from Salmonella, typhimurium and E. coli. So in some of the transgenic plants like cotton, tobacco, potato, there is a particular gene called as AROA gene which is being introduced from the bacteria Salmonella, typhimurium and E. coli. They are taken from this particular bacteria and they are introduced into these transgenic plants of cotton, potato, etc., tomato, and with the help of which they uh, uh, provide resistance for this particular herbicide, that is glyphosate. So these are the examples of the herbicide resistant. Secondly, second advantage what these transgenic plants can provide to us are the insect resistance. These transgenic plants can also be insect resistance. They can be resistant to different type of insects which are harmful to the plants. So Bacillus thuringiensis is a bacteria that is pathogenic for a number of insect species. So if Bacillus thuringiensis DNA is being introduced into these plants, so it will provide the insect resistant, that is no insect will be able to attack to the particular plant or the transgenic bacteria. Its lethal effect is mediated by a protein toxin it produces. This particular Bacillus thuringiensis produces a protein which is toxic to the insects. So the insects cannot attack the transgenic plants. So these are the examples of the insect resistance plants. Then these transgenic plants can also be virus resistant plants. TMV resistant tobacco and tomato plants are produced by introducing viral coat proteins. So viral coat proteins are introduced into these transgenic plants, by the help of which they are resistant to the virus also. Similarly, pest resistant plants also can occur. In this, transgenic plants are developing a resistant specific pest. Transgenic crops containing insect resistant genes from Bacillus thuringiensis. So they are taking the DNA from the Bacillus thuringiensis, and with the help of which they are pest resistant. Okay, now as we know that these transgenic plants are being introduced to the people for consumer use, so let us know what are the nutritional benefits which we get from these transgenic plants. Nutritional benefits. Researchers have introduced 3 genes into the rice, 2 from daffodils and 1 from a microorganism. So researchers and the scientists have developed a new kind of transgenic rice plant. In this rice plant, the transgenic rice exhibits an increased production of beta carotene as a precursor to vitamin A, and the seed is yellow in color. So this transgenic plant, in easy way we can say that this transgenic rice or the transgenic rice plant is rich in vitamin A, and it produces beta carotene with the which can reduce the deficiency of vitamin A. So it is yellow in color. This particular rice plant is yellow in color. That means rice grains are yellow in color. Such yellow or golden rice may be useful tool to treat the problem of vitamin A deficiency in young children living in the tropics. So this kind of rice transgenic plant, it improves its nutritional benefits, and it is very rich in vitamin A, and it is helpful in removing the deficiency of vitamin A in young children and which are living in the hot areas. Okay, a salt tolerance gene for mangroves has been identified, cloned and transferred to other plants. Similarly, a salt tolerance gene, that means a plant which is tolerant to salt. So this kind of gene has also been identified and cloned, and they are from mangroves and they are being transferred to other plants. This is also one nutritional requirement. The gut D gene from Escherichia coli has been used to generate salt tolerant transgenic maize plants. Transgenic maize plants have been created with the help of the gene that from Escherichia coli, that is bacteria, and these genes are salt tolerant. So these are also providing nutritional benefits. Okay, coming to the therapeutic proteins from transgenic plants, what are the proteins, other benefits which you are getting from the transgenic plants? The other benefits are they can withstand long distance transportation. If we are going to carry these transgenic plants from one particular state to another state, then this has longer they can withstand high temperature and humidity and the weather requirements and the weather extremes. So they can be easily transferred. They can withstand long distance transportation, yielding a longer shelf life. They have a longer shelf life. We can keep it for longer periods and use it for longer periods. Then it has also, these transgenic plants have also created green revolution. Okay, now what are the disadvantages which we are getting from these transgenic plants? Transgenic crops have been suggested to cause allergies in some people. Now these transgenic crops are also introduced into the people so that they can consume and get the benefits, but some people are getting allergic to these transgenic plants, so it is not good for all the people. The antibiotic resistance genes placed in these crops have been suggested to cause resistance to antibiotics. So these plants are also resistant to antibiotic. They are not able to respond to particular antibiotic because the antibiotic resistant gene are being introduced into these transgenic plants. So antibiotics are of no use to these transgenic plants. Third point is population being uncomfortable with ingesting DNA that originated from another source like virus or bacteria. People are also getting scared. People are uncomfortable in digesting these crops from these transgenic plants because they are being originated from bacteria, virus, etc. So people are not comfortable. Transgenic crops can cause damage to the environment. The transgenic crops are also producing some pollens, that is pollution, and they can be very toxic for humans. So this is another disadvantage. It expresses a bacterial toxin in its pollen, which is then dispersed over 60 meters by wind. So this bacterial toxin, there is a bacterial toxin in its pollen, and it can travel by the help of the wind up to 60 meter, and which is poisonous for the human beings, and allergic for the human beings actually. Okay, now these are some of the regulatory authorities for transgenic plants. These regulatory authorities control the transgenic plants and help in its consumption of before consumption of the human population. They are being tested and modified. So what are these regulatory authorities? The first one is Department of Agriculture, that is USDA. Second is Animal and Plant Health Inspection Service. So these regulatory authorities are responsible for regulating the introduction, importation, interstate movement and field release of genetically engineered organisms that may pose a plant pest risk. So before introduction into the human population, these transgenic plants are being tested positively by these regulatory authorities. They before any movement, that is from the laboratory to the field, they are being tested whether it is good for the soil or not. Then they also regulate the interstate movement, that is from one state to another state, the movement of these transgenic crops. Another regulatory authority is Environmental Protection Agency, that evaluates the potential environmental impacts, especially for genes which produce pesticides. Now some of the genes produce pesticides also. So these this Environmental Protection Agency, it evaluates what are the potential causes and what are the impacts it is going to provide on the environment. So there are other regulatory authorities also, that is Food and Drug Administration. It evaluates the human health risk if the plant is intended for human consumption. So before human consumption, Food and Drug Regulatory Authority, it checks the transgenic plants whether it is good for human consumption or not. So now the question arises, should we use these transgenic crops? For before using these transgenic crops, it should be well researched whether it is environmentally sound and non-hazardous for the population. Secondly, more research is required in this field to determine the true safety of these plants. Nobody knows the true safety except the scientists and the researchers. So before introduction of these plants into the common people, it should be well researched and safety should be analyzed. At the least, most would agree that the potential advantage of producing crops. Okay, what are the applications of these transgenic plants? The first applications is resistance to biotic stresses. These transgenic plants, they can tolerate the biotic stresses like weather conditions, resistance to abiotic stresses, other stresses such as heat, temperature and all that. Okay, third is improvement of crop yield and quality. Because of these genetically modified crops, this recombinant DNA technology, it has improved the crop yield and also the quality of the crops. Transgenic plants with improved nutrition, they have added the nutritional benefits in the crops, like as we have already discussed, the rice plant, which improves the deficiency of vitamin A. Then commercial transgenic crop plants, uh, transgenic plants as bioreactors. So these could be okay. Now with the applications, with the applications, we are going to end this topic, that is the transgenic plants. Thank you very much. Good day all.